Hey, Biology 108 students. Today, for this video lecture, I'm only going to go over a small little snippet of some of the lecture material from Chapter 9, Mendel's Laws of Inheritance. In addition to this, I put up two other video links, done by the Amoeba Sisters. They're an excellent series of videos out there that explain a lot of the things we're going to be talking about, and they do quite a little bit better than I'm going to do in these video lectures. But one thing I did want to do is go through what is called a dihybrid cross using Mendelian traits. This is an example from your book, and it's a really good thing to get down because it takes Punnett square and kind of takes it to the next level to really talk about how dominant and codominant and recessive traits work out when you're crossing individuals, sexually reproducing individuals over a couple generations. Because most of the time, Mendel and genetics, it doesn't end at just, you have a dominant individual and a recessive individual. It goes down several other generations, and it can get quite messy. So remember that all these video lectures are really here just to supplement and hopefully clarify some of the book reading. Really important to keep up on that reading for the day. And at the end of the week, when you have any questions, please ask them during the final class chats that we'll generally have at the end of the week in this class. So without further ado, let's jump into some Mendelian genetics. Okay, as you can see, what we're going to start off with on the screen is a smooth yellow pea plant and a rough, wrinkly, kind of green pea plant down here. These are the seeds. And this is what Mendel had set up during his experiments. We do know from previous work that this smooth yellow, both these traits are dominant. So they have big R, big yellow, big yellow. So it's a homozygous individual that's dominant at both of these two alleles. Secondly, what we have with our green pea plant, we know green is a recessive trait, so it's little y, and it's wrinkly, which is another recessive trait, so it is little r. Now, when we put these two, when we breed these two plants together that have these phenotypes, we're going to get what we call an F1 generation. So remember, these are our parental generations or our P generations, and we can signify this with a P. And then when we breed these two plants, we're going to get then our F1 generation there. And our F1 generation, if you've been kind of following along with the Punnett square information, you'll understand that this is going to give us, all of our individuals are going to be of heterozygotes now. So they're going to have a dominant gene and a recessive gene. So all the offspring will have this as their genotype. They'll be big R, little r, and big Y, little y. And because if you remember how dominant recessive works, if you look at big R, that means that these individuals are going to all be round because it swamps out this recessive trait here, though they do possess it. And they're all going to be yellow because they're going to have that big Y and a little y, and the big Y swamps out the little y, so they're not going to be green, they're going to be yellow. And so this is a pretty easy set to kind of think about. But what gets really kind of, I think, tricky is that if we take these F1s and breed two F1s together, what's going to happen, especially because we have all these recessive traits down there. So what we're going to do is work out the problem of what happens when we get an F2 generation. So what we need to do is first set up our Punnett square. So when we set these up just like we would have done to get our F1 generation, that gave us all of our heterozygotes for this. So in this, what we're going to have is we're going to take we're going to basically have them go through meiosis. So we're going to get those gametes. And those gametes, when you produce them, what they're going to do is they're going to, again, segregate these genes around. So what we'll list out all of our sperm from our two individuals. So we're going to have R and Y, and big R, big Y. So both our dominant traits, they will get segregated and moved out. We'll have our big R, but a little y this time. And then we're going to have all of our other cross pairs, little r, big Y, and little r, little y. And we'll put that line there, put another line here, because we're going to have our other individual, and we're going to have to take the gametes from that. We can say these are eggs. So in this case, we'll have big R, big Y, big R, little y again. You can see I'm just using the same top row individuals, all of our pair y's. Little r, big Y, and little r, little y. So this will set up our Punnett square in this case. So now what we need to do is start breeding these, essentially understanding what we're going to get. So if we take our first row here, we have all dominants, and we'll just go down the line and breed them and see how they come up. And so this first individual is going to be big R, big Y, big Y. This will be big R, big Y, little y. Next, big R, little r, big R, little r, big Y, little y, big R, little r, big Y, little I. So they're all going to be expressing all those dominant traits. They're all going to be smooth yellow seed colors. So let's go down our next row then. And so here we're going to have big R, big Y, little y. So again, all those dominant traits are going to be expressed. However, when we go down, we're going to start hitting something interesting now. So in this case, we have big R, but now we have two little y's, so two recessive traits. So little y and little y. And remember, our Y genes are color genes. So this is going to be smooth coated. So because we have the two smooths here, and these are going to be green, however. So they're smooth green seeds that this individual is going to possess then. And we can keep continuing down our row again. So big R, little r. So again, they're going to be round. And we have big Y this time and little y. Okay, so again, we have a smooth round individual. Below that, we're going to get a, jump the gun on that one. We're going to have a big R, little r, so they're going to be round. But again, we're going to get two recessives here. So then this individual is going to be green, like we had above. All right, so now we have our last row here. RY will be what we'll start with. So we'll take RY, little r, big Y with big R, big Y. So this will be R, little r, and big Y, big Y. So again, they're going to be a smooth yellow seed. The next one, we're going to get that same thing, big R, big Y, little y. So we have that phenotype there. And then going down, we're going to have two little r's here. So this means that this is going to be a rough individual there. But if you kind of are following along, you'll know that this is still going to be a yellow. So it's going to be a wrinkly yellow individual. And our next one's going to do the same thing. Little r, since you have two recessives, those recessive genes are going to be expressed then. But again, it's going to be yellow because it's going to have that dominant big yellow Y and a little y. All right. So now we have one more row to go, and I'll kind of let you guys predict ahead of me. I'm going to go through this really slow, and so you should be able to kind of catch on by now. So we have big R, little r, and so that's going to be expressed as big R, little r, and then our big Y, little y. So again, that's going to be that smooth yellow individual. Next, we have big R, little r again. So this individual is going to be round, but we have two little y's, so that makes it a green individual. And then we have two little r's, so this is going to be a wrinkly individual. But we have one big Y, so it's going to keep it as being yellow. And then our very last one down here is all recessive traits, so it's wrinkly and it's green. So what I'm going to do, I'm going to go and I'm going to circle all of our wrinkly individuals here. So all those ones with the little r's. And now from here you can kind of start to look at the ratios we have. So we produce 16 offsprings through this dihybrid cross. And the majority of them are going to be smooth yellow individuals, as you can see as we lined out there. In fact, half of them are going to be that. So 9 out of 16 are those smooth yellow individuals. When it came to wrinkly individuals, though, that are yellow, we have 3. So we have this guy, and this guy. Those are the 3 that are that inherited that recessive trait for wrinkliness. And when it came to green ones overall, we only had about 4 greens overall, and 3 of those are smooth because they have those dominant R traits there. And we have one green one, though, that's wrinkly, that has all those recessive traits there. So it's important to remember doing these is to really take your time and consider what is a phenotype versus a genotype. So somebody could have a recessive trait, but not be displaying that recessive traits. And this is one of the reasons, and especially in public health, that's really important for understanding hereditary traits. And it's a good explanation of why something like Huntington's disease and multiple sclerosis, all these really debilitating diseases, are still around in human populations. Because we didn't really understand how this played out in humans, and really until the 1950s, even though Mendel's work was quite old. So practice doing a couple of these, and if you can't get quite the hang of it, maybe you try it during our writing assignment this week. You can let me know, either email me, or you can wait till this class chats and I can go through it again. So anyhow, I hope you're all doing okay, and make sure to check out those other 2 videos I posted and do the reading, and let me know if you have any questions. Thanks, and have a good one.